Good evening. As promised, I'm going to do a lesson on protein synthesis in preparation for the next part of the DNA and genetic diversity booklet. If you've been following along with me so far, we've done the structure of DNA and DNA replication. And there are some pieces between DNA replication and protein synthesis. So a little bit on DNA, genes and chromosomes, and the genetic code, etc. Now, you just need to have a read through that and look at some of the key features, work out what they are. So this idea that the genetic code is degenerate, that there are stop codons, there's non-overlapping and universal, etc., where you can do that at your own leisure. What we are going to focus on today, or in this session, is firstly the structure of different forms of RNA, because they all play an important role in protein synthesis, and then the processes of transcription and translation, which come later. Okay, so let's find structures of RNA. So to begin then, there are three forms of RNA. They are mRNA, or messenger RNA, tRNA, which is transfer RNA, and finally rRNA, which is ribosomal RNA. Now I'm a little bit picky. Can you please make sure that your M, T and R are all lowercase and that the RNAs are all uppercase, because that's how it should be written. Okay, so we know from previous lessons that all nucleotides have the same three parts. So there is an inorganic phosphate, that's bonded to this five-sided, sugar. That five-sided sugar in the case of RNA is ribose. And then there are four nitrogenous bases. Okay, so these are nitrogen-containing organic bases. And in RNA, they are adenine, guanine, cytosine, and finally, and if you've watched the previous video, you'll know this already, uracil. So uracil has replaced or replaces thymine. Okay, so the nucleotide, RNA nucleotide, consists of those three parts: inorganic phosphate, ribose sugar, nitrogenous base, and those nitrogenous bases are adenine, or A, guanine, G, cytosine, C, and uracil, U. Uracil is different, it replaces thymine in RNA. Okay, so the three forms of RNA, all of them are single-stranded. Okay, so the mRNA, tRNA and rRNA are single-stranded. In fact, the bit that we're most interested in is the mRNA and the tRNA being single-stranded. mRNA, let's focus on that to begin with. Let's try the nice. Let's go for pink. mRNA is a single strand that forms a linear molecule. So a single-stranded linear molecule with nitrogenous bases exposed like so. And those nitrogenous bases are complementary to the bases present on the template strand of DNA. So during transcription, we'll look at this later on, the mRNA forms and those bases, it could be any of adenine, guanine, cytosine and uracil, but they will be the complementary, so the bases are complementary. Let's use a thinner pen here, shall we? Complementary to the DNA. And they're complementary to the DNA template strand. Now we'll look at that in a bit more detail in a moment. Okay, they are a product of a process called transcription. I'll talk a bit about that a bit more detail later. They move from the nucleus, out of the nucleus through the nuclear pores and into the cytoplasm, where they reach a ribosome where translation takes place. Okay, the next RNA that we're interested in is tRNA, transfer RNA, and I always think of transfer RNA as a bit like a taxi service. Okay, it transports, or it transfers, an amino acid to the ribosome, and it has what's called an anticodon, which is complementary to 3 bases that make up the codon on mRNA. We're going to go into this in a bit more detail later, but you'll notice over here, here's the structure of tRNA. It's referred to as being clover-leafed or clover-leaf shaped. Whether or not it looks like a clover leaf is up to you. It has on its as part in its structure a region where I'm not drawing this particularly well. A region where an amino acid joins. This isn't great, is it? Up here, and this is called the amino acid binding site. And that amino acid binding site is specific to 3 bases that stick out at the bottom down here. And these 3 bases are referred to as the anticodon. And the anticodon is complementary to 3 bases. For example, these 3 on the mRNA. So that's the anticodon. mRNA contains codons. Okay, those codons are complementary to the triplet codes on DNA. Now I'll come back to that in a bit more detail later. Okay, then the final rRNA. The only thing you need to know about rRNA is that it forms part of the ribosome. So rRNA, or ribosomal RNA, oopsie daisy, a ribosome contains, or we can see it down here, a ribosome formed from rRNA and protein. And that's all you need to know for that. One other thing, sometimes this confuses people. tRNA is single-stranded as well. You can see it's actually one continuous single strand, but it's folded into this clover leaf shape. Folded into the clover leaf. And it's held in position by hydrogen bonds, and those hydrogen bonds just form between different nucleotide bases, nitrogenous bases, within the same RNA molecule. Okay. Yeah, have I missed anything out there? No. The only thing, just to reiterate, that anticodon determines which amino acid is able to bind at the binding site. Okie doke. So, differences between mRNA and tRNA is a fairly common question that you might get in an exam. If you're asked about the differences, it says here about these exam tips, and this goes for any question where you're asked to give differences, or if you're asked to contrast. It's important that you give both sides of the comparison, so you make a comparative statement. So we say that mRNA is linear, whereas tRNA is clover-leafed. Okay, so we've got that whereas, making it comparative. The mRNA contains codons, or possesses codons. tRNA possesses an anticodon. No hydrogen bonds present in the mRNA, whereas there are in tRNA. No amino acid binding site in mRNA, whereas the binding site obviously exists on the tRNA. Sorry, no amino acid binding site on mRNA. There is one on tRNA. Okay, right. Let's move on to transcription then, and the beginning of protein synthesis. So you have this diagram in your notes. It summarises transcription and translation, the two processes that are involved in the formation of a protein, in protein synthesis. Okay, so in the nucleus we find DNA. DNA is transcribed into mRNA. Now transcription, or to transcribe, means to copy. So by that we mean that the DNA is copied into mRNA. We can think of the DNA and all of those chromosomes a bit like a library. So it contains lots and lots and lots of information, and it wouldn't be possible to take all of that information out all at once. And mRNA is a bit like a book, so it's a smaller part of the information held within the library, if we're going to continue that analogy. And that mRNA can be taken out of the library, or really out of the nucleus, and into the cytoplasm. So DNA is copied into mRNA. By copying we mean we remain in the language of nucleic acids. So DNA and RNA are both nucleic acids, so we've just copied from one into another. Then at the ribosome, that mRNA binds to the ribosome, and amino acids are carried to the ribosome by the transfer RNA. Remember I said the transfer RNA is a bit like a taxi, so it picks up amino acids, drops them off at the ribosome, and then goes to pick up more. And here the second process takes place, translation. So as we know, translation means to change from one language into another. So here, the DNA and the mRNA are the language of nucleic acids, or it's like a genetic language. Translation takes that genetic language, those language of nucleic acids, translates it into the language of polypeptides, or sequences of amino acids. Transcription, copying, translation means that translating from a genetic language into polypeptide language. A couple of really important key terms here. So genome means the complete set of genes within a cell, and that determines the proteome. So the proteomes are all of the proteins that a cell can produce. Right then, so you have this description in your notes. Okay, I'm going to try to draw a diagram to show what is happening, but you've also got this one here. So let's see if I can do a diagram. Okay. Okay, so DNA is double-stranded, and we know that the DNA contains those bases which hold the genetic code. So the bases like this. So those bases form on the DNA. So held in place by the sugar phosphate backbone, those inorganic phosphates, and the deoxyribose sugars held by the phosphodiester bonds. Four, five, six, seven. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7, 8, 9. 8, 9. Okay, so we're going to call this strand here the template strand, and we're going to call this one here the sense strand. Okay, so what happens in the nucleus then? So if we have a look over here, so in the nucleus we get this rewriting or transcribing of the DNA into a strand of messenger RNA. The relevant section of the DNA uncoils, so that means this gene, we're going to call this the gene, and this is the gene which will be transcribed. It codes for a specific sequence of amino acids which make up a polypeptide. One of the strands acts as a template. This one is going to be the template at the bottom. And individual RNA nucleotides line up alongside the exposed bases. So we're going to call these over here, these are the exposed bases. Okay. So actually there is a complex of proteins that separates those two strands by breaking the hydrogen bonds. Now, you don't need to refer to those, but it has a similar role to DNA helicase that we spoke about during DNA replication. This forms those exposed bases. So those are the exposed bases on the template strand. Now, just like in DNA replication, we get complementary base pairing. Okay, so 3 RNA nucleotides pair up with their complementary base pairs on the template strand. So these are the 3 RNA nucleotides. They pair up by complementary base pairing on the template strand, and that is spontaneous. They pair up because of the complementary base pairing. Then an enzyme joins those sugars and the phosphates together. It's a bit thick, isn't it, that one? So an enzyme moves along and joins those sugars and phosphates together, forming phosphodiester bonds. That enzyme is called, let's make it nice and big, let's draw that enzyme over here. That enzyme is going to continue in moving in this direction as the 3 RNA nucleotides pair up with their complementary base pairs on the template strand of DNA. This enzyme will join together the 3 RNA nucleotides to form that sugar phosphate backbone by forming phosphodiester bonds. Now this enzyme is known as RNA polymerase. Okay, RNA polymerase, its function is to join RNA nucleotides, so it forms the phosphodiester bonds. Just like DNA replication, those nucleotides line up spontaneously due to the complementary base pairing. The enzyme, RNA polymerase, then just joins the nucleotides together. Okay. Now, once it reaches the end of the gene, or the end of this piece of DNA that's due to be copied into mRNA, the mRNA detaches from the template strand and actually at that point it leaves, let's see if I can draw this. This is the nuclear membrane. Okay, it leaves the nucleus and moves into the cytoplasm, where it will bind to a ribosome. Now, in eukaryotes, well, this is all a eukaryote if this contains a nucleus, but in eukaryotes a process happens before. So what's going to happen is this mRNA will leave the nucleus. But there's a little bit more going on in this part here, and we'll just revert back to the PowerPoint to show that. Okay, we call this, where are we? Splicing. Okay, so you may know this already, but after the Human Genome Project was completed, we found that about only roughly 2% of the human DNA codes for proteins, and that was really unexpected. There are a lot fewer proteins actually produced, there are a lot fewer genes than were expected, and most of the DNA, a massive percentage of the DNA, doesn't code for protein at all. Okay, and the reason for that is because there is lots of non-coding DNA. So, non-coding DNA just means it doesn't code for a protein, and they include within the genes, so within the base sequence, the nucleotide base sequence of the DNA, these regions called introns. Okay, introns are within, and they are non-coding. Okay, you must refer to them as introns and not just non-coding DNA, because there are also, between the genes, multiple repeats. Multiple repeats are areas where maybe one or two nucleotide bases are repeated hundreds, possibly thousands of times. But those introns, which are non-coding, are copied across into are copied across into the mRNA. So the first molecule of the mRNA that is produced is known as this is pre-mRNA. And pre-mRNA contains the introns, which are non-coding, and exons. So I always think of exons as being expressed into proteins. Let's go back to the PowerPoint. Okay, so we can see those exons here. Now the exons are the bits that do code for proteins. The introns are in between them, and they need to be cut out. So they need to be removed from the pre-mRNA. This shows a multiple repeat called a tata repeat. You can look that up if you want. This shows the exons. Okay, so here's the DNA with exon, intron, etc. Only those exons are passed on to the mature mRNA. That will code for the protein in the end. So the introns, these regions, need to be removed. And they are removed in a process called splicing. Splicing happens like this. Okay, splicing, or sometimes referred to as post-transcriptional, so after transcription processing. Here is the template strand of DNA, exon, intron, etc. The pre-mRNA, exon, intron, it's just a copy of that. Now those introns must be removed, okay, leaving the mature mRNA, which contains exons only. Okay, the mature mRNA, exons only, and it's the mature mRNA that leaves the nucleus through the nuclear pore. So what we've got over here, pre-mRNA gets formed first, the introns get removed, leaving the exons only, and this is mature. It's a bit untidy, this, isn't it? This is the mature mRNA that leaves the nucleus through these nuclear pores. Goodness. Right, nuclear pause. And that is transcription. Now I'm going to stop the video there because uploading massive long videos takes so long on YouTube, and I'll be back with translation in a moment. So